Let's go over the enzymes involved in DNA replication. Remember that DNA replication is the process of copying DNA and producing two identical strands of DNA. And this process occurs during the S phase of interphase. Our first enzyme that we're going to talk about is called topoisomerase. This is an enzyme that's going to be involved in untwisting or unwinding the DNA. You can see topoisomerase working ahead of helicase and just unwinding the DNA. Our next enzyme is called helicase. And this enzyme is going to break the hydrogen bonds between the two strands of DNA. So basically this enzyme unzips the DNA. You can see the enzyme helicase here unzipping and breaking the bonds between the two strands of DNA. These next proteins aren't technically enzymes. They're called single-stranded binding proteins. And they're going to help make sure these single strands of DNA don't get tangled up and get damaged during this replication. These single-stranded binding proteins are also called SSBPs. Our next enzyme is going to be called primase. Primase is going to place an RNA primer on the DNA for DNA polymerase to bind with. So basically, primase is going to show DNA polymerase where to start. You can see primase leaving an RNA primer for DNA polymerase to bind with and initiate replication. And this leads us to our last but most important enzyme, DNA polymerase. This enzyme is going to find and bind free-floating nucleotides to their complementary base pairs on the free strand of DNA. You can see DNA polymerase binding these free-floating nucleotides on in a 5 to 3' direction. Now it's important to remember that this DNA polymerase runs along the 3 to 5' direction. However, this DNA polymerase makes the DNA in a 5 to 3' direction. I almost forgot our last enzyme, ligase. This enzyme is going to work on the lagging strand. It's going to connect the fragments of DNA that don't have a complete bond. So basically this enzyme is just going to bind DNA fragments together.